Binary fission is the asexual reproduction by a separation of the body into two new bodies. During this process, the organism duplicates its genetic material, which is known as DNA, and then divides into two parts. At the end of this process, each new organism receives one copy of DNA. Each daughter cell is identical to the parent cell. So who uses binary fission? Binary fission is primarily used by prokaryotes. However, it does occur in some single-cell eukaryotes. I want to start out by giving some characteristics of prokaryotes. Prokaryotes are unicellular, they lack a membrane-bound nucleus, and they lack membrane-bound organelles. They are also classified into two separate domains, bacteria and archaea. Because their cell structure is so simple, they can only reproduce asexually through binary fission. There are 5 steps to binary fission. Step number 1 is DNA replication. Number 2 is cell elongation. Number 3 is the septum starts to form. Number 4 is cell is partitioned. And finally, number 5 is daughter cell separates. Just like in human cells, bacteria needs to copy its DNA. However, bacteria cells usually have singular, circular chromosomes that always lack a nucleus. The bacterial chromosome is found in a specialized region of the cell called the nucleoid. The copying of DNA by replication begins at the origin of replication on the chromosome. The origin of replication is close to the binding site of the chromosome at the plasma membrane. The chromosome is uncoiled and copied to form a new chromosome, thus doubling its genetic content. This is the very first part of DNA that is copied. Step number 2 is cell elongates. The replication of DNA is bidirectional, meaning that as the cell continues to grow and new double strands are formed, the two origins move towards the opposite ends of the cell. As the origins move to the opposite ends of the cell, it pulls out the rest of the chromosomes along with them. This movement causes the cell to elongate, which only adds to the separation of the newly forming chromosomes. As this is happening, DNA continues to be copied. Step number 3 is the septum starts to form. Once the new chromosomes have moved to the opposite cell ends and have each cleared the midpoint of the cell, division of the cytoplasm starts to take place. The cytoplasmic separation contributes to the formation of a ring, which is composed of repeating units of the FTSZ protein. This will help direct the partition between the nucleoids. The formation of this FTSZ ring stimulates the accumulation of other proteins that work to gather new membrane and cell wall materials at the site. The septum is now formed between the nucleoids. Step number 4 is the cell is partitioned. When the new cell walls are in place due to the formation of the septum, the cell splits itself down the middle. Step number 5 is daughter cells separate. The two daughter cells are released as individual cells. These cells are identical to the parent cell. They each contain the same nuclear material and cell organelles. This process happens for every new cell, which leads to growth for the population.